In this video, I'll be talking about sequence analysis of 16S rRNA to identify the bacterial species. Suppose you have already done the 16S rRNA PCR, checked the amplification in agarose, got the amplified product, cleaned up the gel and finally purified the product. Then you give your PCR product for sequencing or you have run the sequencing. At the end, you will have the sequencing result which further you need to analyze to find out the name of the bacteria. So here, suppose we have two files, forward and reverse sequence. You can analyze either using the snap gene or Finch TV. Here I am using the snap gene to analyze. This is how the interface of snap gene looks like. This is the 27F forward sequence. Take the forward arrow. Here you can see peaks of different color. Here each peak indicate one base. Suppose here you can see G is represented by the black color, A is by green, C is by blue and T is by red color. And in the bottom, quality of the peak is shown. This is the region where primer binds, so the quality of the peak is not that good here. You can see after the primer region, we are getting good peaks. So now we have to copy the peak region from the region where primer ends and we are getting good peaks to the end region of clean sharp peak. Usually this will cover 750 to 800 base pair from the primer. So now we have copied the peak area and now we have to paste the sequence in the Word file. Here we have pasted the forward sequence. So now we have extracted the DNA sequence for the forward primer. Now same thing we will do for reverse sequence. First we have to open the reverse sequence file using snap gene. Here the primer region is present at the right side of the sequence. First just take the reverse arrow and copy the region from primer region to the end region where you are getting good peaks. So here we have to start from the middle of the sequence around 800 to 850 base pair. So now we have to copy the region from middle to the end just before the primer. Paste the sequence for the reverse primer in the same Word file. So now you have both forward and reverse sequence. Select few bases from the starting of the reverse sequence and now copy and press Ctrl F and paste the sequence. Because when you align forward and reverse sequence, you will get overlapping regions in the middle. Here you can observe that in the forward sequence, after the yellow region, base pairs are same which are already present in the reverse sequence. So remove the extra sequence from the forward primer. Now bring the cursor in front of the reverse sequence and press backspace. Now you have got the final sequence of the DNA. You can use this sequence to identify the bacteria. Now in the browser, type EasyBioCloud, select the easybiocloud.net and fill your login ID password. This site is free, you just need to sign up first to use this tool. After sign up, select 16S based ID, then paste your sequence and submit this sequence. After few seconds, you will see the result. You can see the similarity of your sequence to the available database. EasyBioCloud performs a sequence alignment of the input data against the reference sequences in its database. This alignment involves comparing the query sequence to known sequence to identify similarities and differences. Like here, you can see the input sequence shows the similarity with the bacteria already present in the database. Now, you can also identify the bacteria using NCBI nucleotide blast. You just have to paste your sequence and blast it. This will also show you the similarity result of the query sequence to the database. So this is how you can identify the bacteria. I hope you liked my video. Please like, subscribe and share with your friends. Thanks for watching.